Hello friends, I'm Vishal Patel. I'm working as a lecturer at HVHP Institute of Postgraduate Studies and Research. Today I'm going to present my presentation on microbial analysis of water, and that is based on the pharmaceutical manufacturing and sterility testing. This is the topic of pharmaceutical technology, MSc Semester 3 Microbiology. So, first thing, why water to be tested? Water is widely used in pharma company for manufacturing of the different products, as water is most important ingredient in manufacturing. That's why it is necessary to be tested here. It contains microorganisms that can be harmful or pathogenic. So mainly for catching of the pathogenic organism, water to be tested is very important. After the completion and the separation from the raw water, it is defined and separated and categorized into the four different types of the water, categorized in purified RO, water for injection, and the pure steam. Pure steam is generated from the water for injection. Rest of three, purified RO and the water for injection, is essential and very important parts and what important water types in pharmaceuticals. First thing is sampling procedure. If we want to do microbial analysis, we have to do the sampling at first. And for sampling, analysts should know all rules and regulations regarding good manufacturing processes and the water sampling. Water sampling done in sterile bottles along with the 70% isopropyl alcohol. Your bottle should be sterile before sampling. All sampling point must be clean before taking samples, and it must be drained up to 1 minute to reduce the chances of the contamination or pathogens in water. Analysts have to do this activity in daily or in shift basis. Types of the testing. Water testing is divided into the two parts. Two methods are there. First one, membrane filtration method, and second one is a microbial enumeration test for the specified microorganisms. These two methods having a different and distinct features. First one, membrane filtration method. You can see in the first picture, this is the membrane, cellulose membrane, and the pore size is 0.45 micrometer. Second picture contains the apparatus for the membrane filtration method. You can see this contains a sample holder simple to hold the sample container and the vacuum flask. The vacuum flask is connected with the vacuum so that can suck the water from the sample container. In the third picture and the fourth picture, these are the pictures of the manifold which is made up of the magnet. So you can taste more than one samples at a time. So these are the pictures of the membrane filtration. The procedure of the membrane filtration is like you have to take 100 ml water for the one cycle of the water testing. With the help of the cellulose membrane, as I said, 0.45 micron pore size, we can trap microorganism from the water. Vacuum can suck the water from the container to vacuum flask, and after completion of the 100 ml water, membrane then placed onto the R2A agar. What is R2 agar? R2 agar is a minimal nutrient medium which is made to grow oligotrophic organism. Mainly in the water, there is no nutrient. The oligotrophic organism can grow into the water. And if we want to catch the microbial flora that are oligotrophic, so we have to use the R2 agar. Incubation and the observations. This method is a very simple method. We have to incubate it at the 30 to 35 degrees Celsius for the 5 days. Plates are observed by the colony counter. You have to mark one colony, and then this table shows the limitations for the different water. You can see the potable water, purified water, and the water for injection. First limit is alert limit, and second limit is action limit. Both are for the CFU per ml, colony forming unit. You can see the potable water, purified water, and the water for injection. The alert limit is 250 and 0. Water for injection contains 0 colony, while potable water containing 200 colony. That is the alert limit. That means alertness of the colony forming unit, your bacteria. If your bacteria or CFU is exceed from the alert limit, that is categorized into the action limit. That means you have to take actions towards the water sanitization system, and that is, you can see 300, 75 and 1. Second method, most important method, is microbial enumeration for the specialized organism or pathogenic organism. This test is most important to check whether pathogens are present or not. This is done for the four organisms: Escherichia coli, Pseudomonas aeruginosa, Staphylococcus aureus, and the Salmonella enterica. E. coli, Escherichia coli, and the Salmonella enterica are the coliforms, while Pseudomonas gram-negative and Staphylococcus aureus is gram-positive cocci. Media. You can see there are so many media enlisted, starting from the soybean casein digest medium, that is a simple medium like nutrient medium, nutrient broth, MLBB, McConkey lactose bile broth for the E. coli, McConkey agar for the E. coli, eosin methylene blue agar for the E. coli, mannitol salt agar for the Staphylococcus aureus, cetrimide agar for the Pseudomonas aeruginosa, repaprot vacillati salmonella enrichment broth, and the xylose lysine deoxycholate agar for the Salmonella enterica. So these are the different media for the different organisms. Starting from the Escherichia coli. So the first procedure is same for all four organisms. For that, you have to take 1 ml water and add or inoculate into the SCDM broth, soybean casein digest broth, and have to incubate for the 24 hours at 30 to 35 degrees Celsius. On the second day, you have to take a loopful and transfer into the MLBB broth, McConkey lactose bile broth. You have to incubate at the 42 degrees Celsius for the 24 hours. If lactose fermenting colony is growing, then second day you have to transfer the loop on the McConkey agar to check lactose fermentation or non-lactose fermentation. Again, you have to incubate for the 40 degrees Celsius for the 24 hours on the third day. On the fourth day, if lactose ferment colony is observed, then you have to transfer this loopful colony to the eosin methylene agar to check whether it is E. coli or Enterobacter. If metallic green shine is shown, that confirm that your water is containing Escherichia coli. Second is the Staphylococcus aureus. Same procedure. For this organism, you have to add 1 ml water sample to the SCDM broth and incubate for the 24 hours at the 30 to 30 degrees Celsius. Second day, you have to take a loopful from the SCDM and have to transfer onto the mannitol salt agar. Mannitol salt agar is a specified medium for the Staphylococcus aureus. Then you have to incubate for the 24 hours again to the 30 to 35 degrees Celsius. And third day, if red color colony observed, that confirms the Staphylococcus aureus growth. Many pharma companies do the rapid test for the Staphylococcus aureus because Staphylococcus is a very dominant and the potent organism that is present in the water. So rapid plasma test is a rapid test that has done for the Staphylococcus rapid test. Third one, Pseudomonas aeruginosa. For this organism, first you have to add 1 ml water sample to the CDM broth. You have to incubate for the 24 hours at the 30 to 30 Celsius. Then second day, you have to transfer loopful from the SCDM to the cetrimide agar. Cetrimide agar is a specified medium for the Pseudomonas. Again, you have to incubate for the 24 hours and the 30 to 30 degrees Celsius. And third day, you have to observe the result. If the Pseudomonas colony is grown on the cetrimide agar, it confirms that Pseudomonas is present in your water. Fourth one, Salmonella enterica. For this organism, you have to add same 1 ml of the water sample to the SCDM broth. You have to incubate for the 24 hours at the 30 to 35 degrees Celsius. Second day, you have to transfer a loopful to the Repaprot vacillati salmonella enrichment broth. This is a blue colored specified broth, specialized media, differential media for the salmonella. Then you have to incubate for the 24 hours at the 30 to 35 degrees Celsius. On next day, medium color shows the result indication. If your media is darker blue, that indicates your salmonella is growing. Then you have to take a loopful and transfer onto the XLDA, xylose lysine deoxycholate agar, for the confirmation of the salmonella. Next day, you have to observe the result on the XLDA with the black colored colony with the smell of the H2S. That indicates a growth of the Salmonella enterica. What will be the conclusion after these four organisms? All tests should or must be negative, means no pathogens allowed in water. So water must be free from pathogens which will be used for the pharmaceutical manufacturing. Our next test is the sterility testing. This is very important testing among all the tests that is done in pharmaceutical microbiological analysis. This test is done for the parenteral product and the medical devices. Okay, this is a particular test that is categorized only for the parenteral products and the medical devices. You cannot do a sterility testing for the solid doses, okay, like tablet and the capsules. This is why this is sterility testing is important, because without the result compliance of the sterility testing, no product can dispatch to the market. You have to submit the document of the sterility testing for the compliance, then and then that product can or will dispatch into the market. So what exactly this sterility testing? The test for sterility is carried out under the aseptic condition. In order to achieve such conditions, the test environment has to adapt to the way in which the sterility test is performed. The precautions taken to the avoid contamination are such that they do not affect any microorganism which are to be revealed in the test. The working conditions in which the tests are performed are monitored regularly by the appropriate sampling of the working area and by carrying out appropriate control. The following culture media have been found to be suitable for this sterility. First one is a fluid thioglycolate medium. Is a primarily intended for the culture of the anaerobic bacteria. If your product is containing any anaerobic organism, that will be catched by the fluid thioglycolate medium. Second, however, it will also detect the aerobic organism. Soybean casein digest medium is suitable for the culture of the both fungi and the aerobic bacteria. If your aerobic bacteria and fungi is present in your product, that will be catched by the soybean casein digest medium. Fluid thioglycolate medium is to be inoculated or incubated at the 30 to 30 degree Celsius for the product containing the mercurial present preservative that cannot growth promotion test, and the method suitability test be tested by the membrane filtration method. Fluid thioglycolate medium incubated at the 20 to 25 degree Celsius may be used instead of the soybean casein digest medium, provided that has been validated as described in growth promotion test of aerobes, anaerobes, and the fungi. Growth promotion test I talk to the later, where prescribed or justified and authorized the following alternative. Thioglycolate medium might be used prepared a mixture having the same composition as that the fluid thioglycolate medium, but omitting the agar and the resazurin sodium solution. This is the composition of the both media. You can see the fluid thioglycolate medium that containing the resazurin sodium solution, and that create a one ring, red color ring on the top of the media. Okay, soybean casein digest medium is a simple medium, as I earlier said into the water testing. So this both media are the heart of the sterility testing. Now, this is the table of the growth promotion test. Growth promotion test, that means you have to check your media with the use of the ATCC cultures microorganisms. Means if you want to do soybean casein digest medium testing, first you have to check the medium suitability. Means your media is able to grow organisms, that and then we can use this media for the further testing. If organisms are not able to grow on that media, growth promotion test is failed, and that media is not further used for the testing. So you can see here different strain for the test organism suitable using the growth promotion test or in method suitability test. Staphylococcus aureus, Bacillus subtilis, Pseudomonas aeruginosa, anaerobic organism, Clostridium sporogens, fungi Candida albicans and Aspergillus brasiliensis. These are known as a reference organism that is used for the growth promotion test. Growth promotion test is a very basic test that is done while you are making different media. On the day of the media making, you have to do the growth promotion test. If you are making media for the sterility testing, you have to do growth promotion test for the sterility media. Types of the sterility testing. There are two types of the sterility testing is that membrane filtration method and the direct inoculation. This will be varying to the depend upon the nature of the product. Some diluent are used in the membrane filtration method to nullify the chances of contamination like peptone water, beta lactamase, and cephalosporin and polysorbate 18. Means if you are doing a different product, if you are using different product to nullify the chances of contamination, you have to use some diluent. Peptone water also used. This is the chamber of the sterility testing. You can see this is the manifold. You can see here manifold. You can do analysis of more than one sample, and here you can put one cellulose membrane. What you have to do after completion of the sucking or adding of your media of your product, you have to cut that particular filter paper or membrane into the two piece. One piece you have to inoculate in the soybean cage in digest medium, and one piece you have to inoculate into the fluid thiogolate medium. Okay, the amount is varying from the product to product. Here, this is the minimum quantity to be used for the each medium. As I said, different solution or product having a different capacity for the sterility testing. Here you can see the liquids for the less than 1 ml, 1 to 40 ml. This is the quantity that is made by the pharma companies. Greater than the 40 ml, greater than 100 ml, greater than 100 ml. Antibiotic liquids for the solids. Okay, you can see this chart. So as per the different product, you have to do the different sampling or different lot from the batch. Incubation period. 14 days total time for the incubation for the sterility testing. It will take 14 days. 14 days are the too long. 7 days for the 20 to 25 degrees Celsius and 7 days for the 30 to 35 degrees Celsius. Both having a different nature. So if your microbial flora, that is fungi or aerobic organism, that will be grow into the 20 to 25 degrees Celsius. If your anaerobic organism is present in your product, that will grow into the 30 to 35 degrees Celsius at interval during the incubation period, and its conclusion examine the media for the macroscopic evidence of the microbial growth. If the material being tested renders the medium turbid, so that the presence or the absence of the microbial growth cannot be readily detected by the during the 14 days, if you find any one test tube that is turbid, your sterility testing is failed. And there are so many investigations during the sterility testing. If your product is failed in sterility testing, the analysts have to do the again that product sterility testing and examine under the 14 days. If next 14 days your product is not able to give the sterility testing compliance, then they have to do the investigation with the pathway of the testing with the use of the SOP. The reviewer will see the testing of the analyst. These are the rules and regulation, and these are the steps of the SOP if failure is done. So 14 days after the beginning of the incubation, transfer portions is not less than 1 ml of the medium of fresh vessels of the same medium, and then incubate the original for the vessels for not less than 4 days. If no evidence of the microbial growth is found, the product to be examined compliant with the taste of the sterility. If evidence of the microbial growth is found, the product to be examined does not comply with the taste for the sterility, unless until it can be clearly demonstrated that the taste was invalid for the causes unrelated to the product to be examined. The taste may be considered invalid only if one or more of the following conditions are fulfilled. These are the different conditions that is followed for the your test is positive or negative. The data of the microbiological monitoring of the sterility testing facility show a fault. A review of the testing procedure used during the test um, in questions revealed a fault. Microbial growth is found in a negative control, and after determination of the identity of the microorganism isolated from the taste, the growth of this species may be ascribed unequally to the fault. These all things contain each and every aspect. You can see for the first, during sterility testing, you have to do the environmental monitoring also of that particular room. Means saddle plate and the air sampling you have to do along with the sterility testing. So we can check the room healthiness of that particular sterility clean room. Your room is strong enough aseptic, having the aseptic environment or not, that is checked by the environmental monitoring while doing the sterility testing. Second one is a review of the testing procedure used during the test in a question revealer fault. If analyst having the some problem regarding the testing, so that is the one type of the fault. Microbial growth is found in negative control. As I said earlier, you have to do the growth promotion test. So we are putting positive control also. But this is for only the media suitability. If your negative control is going to be contaminated, so that is a fault that is caused by the analyst. So this is a one type of fault. Okay. So these are the different um, these are the different fault that cause our sterility test as invalid. In the test is declared to be invalid, it is repeated with the same number of the unit as in the original test. If no evidence of the microbial growth is found in the repeat test, the product growth is found in product examined complies with the test for the sterility. If microbial growth is found in the repeat test, the productive examined does not comply with the test for the sterility. So this is very critical test also, because 14 days is large number, and if your test is not going to pass or comply, you have to do the further testing for the 14 days, and it will take this is a critical time for the pharmaceutical company also, because many products are stuck with this sterility testing. First, it will take so much time, because some tests are for the 1 days or 5 days. Microbiological testing are longer compared to the chemistry and other tests. So this is very important test, and this indicate complications and the compliance of the total pharmaceutical asepticness. Your system is healthy or not, that is detected by this sterility testing. Your handling is strong or poor, that is catched by this sterility testing. Thank you so much.